Hello students. So today we are going to look at the synopsis of the chapter Principles of Inheritance and Variation. So I hope you have already studied this very important chapter from the point of NEET in detail. This chapter carries a lot of weightage in NEET as well as in the board exams. So let's quickly have look at the summary of this chapter. Now, you have already studied about what inheritance is. It is the process by which the heredity is passed on to the next generation. So, whatever the character is, that is passed on from one generation to the next generation. Now, looking at the first point, resemblance and variation. In the right side, you can see this image of two young girls, and you can see the resemblance in them. You can see that they are twin girls, and they actually resemble each other. They are alike each other. So this is also due to the inheritance. One character is passed on from the next generation. But if we talk about the variation, here you can see that each individual is different. So there is a lot of variation to be seen in this image. And this also comes as a part of the inheritance, passing on of one character from one generation to another generation. We'll also have a look at the terminologies, the very important terminologies that you've already studied in this chapter. You have studied about the gene, that is it is also called as the factor by the Gregorian and Mendel. So this is the one which is the basic unit. Okay, it is the basic unit of heredity. We also talked about the traits. So traits are basically the characters. Your eyes are black in color because your parents, one of your parents, maybe your mother or your father had black colored eyes. And that's why the character is passed down from one generation to the next generation. Talking about the gene locus. So gene locus is basically, locus means what is the exact position of that gene. So if you look on the chromosome, wherever that gene is located is called as the gene locus. Allele are basically the alternating forms of gene. So you can see that there can be different alternating forms of a gene. As you can see over here in this particular image, there's capital A and small a, the small a and capital A. So in this particular image, you can see the gene locus is changing. First, the small a is here, and the second image, the small a is here. And similarly, vice versa with capital A. So this is the gene locus, wherever the gene is located. But we can also see that it has two alternating forms. We'll study about it, dominant and recessive. Homozygous and heterozygous. So homozygous means that if that person contains the genes, both the alleles are alike. Both the alleles are alike. That would be called as the homozygous. So in this case, you can see the pink and the pink. Okay, so both the alleles are alike, so that is homozygous. So this particular chromosome is homozygous for that particular allele. Whereas here you can see the yellow and the green one. This means that they are basically heterozygous. Heterozygous for that particular allele. So the both the alleles are different. Here you can see the yellow and the green. Then there's phenotype, and then we'll also study about the genotype. So phenotype is basically the character that is mostly visible, okay? And the genotype is the one which is present in the genes. So here in this image you can see, so this is what is present in the genes or in the chromosome. And we can't actually see it. Whatever is visible to us is what will be the characteristic of that particular insect. So what is visible is called as the phenotype, and whatever is not visible is present in the genes will be called as the genotype of the individual. Gamete, we've already studied, we already know about gametes. So gametes are basically haploid, so basically they are secreted, or they are as a result of division of the cells. And whenever division of the chromosomes takes place, and mostly into half, that results into a gamete here. So you can see in the cell, in the parent cell, you can see that four chromosomes are present. But here in the daughter cell, you would see that actually only two chromosomes are present. So that way it is a gamete. And to remember, the gametes are always formed in the sex cells. Yes? Now moving on to what is a hybrid. See, one generation, one parent, interacting with another parent of, maybe some other quality, and then forming another species which is a hybrid of both, containing the character of both the parents, that will be called as a hybrid. Yes? Okay. Now moving on to the next terminologies, F1 generation is the first generation. So when the parents have, fused, and the first generation that is formed is called as the first filial generation. That is why it is called as the F1 generation. First filial generation. And if we talk about the F2 generation, if these reproduce, okay, so this F1 generation, again, they undergo self-film and they produce some flowers, these would be called as the F2 or the second female generation. Yes, you've already studied about that. Okay, moving on to what is a dominant allele. See here in the example, you can see dominant allele is basically those alleles whose characteristic is expressed in the next generation. Yes. So here, these are the parental generation. This is the F1 generation. And in the F1 generation, you can see that this particular crane is pinkish in color. And pink is basically this particular parent. Suppose consider this is the female and this is the male. So the pink color is coming out of the female. That means this particular allele is a dominant allele. Whereas if we talk about recessive allele, see this purple color, violet color didn't get expressed in the F1 generation. Okay, so this particular allele is going to be the recessive allele. Okay, I hope that is clear. We are anyway just going through whatever we have studied. Then there is Punnett square we talked about. In Punnett square, what we used to do, as many characteristics we had, we used to create a square of them, as many genotypes we had. And then from that genotypes, we used to calculate. Suppose we have capital T small t and capital T small t available to the mother, then in that case, we would fill up this square. So that makes us an easy calculation for us to calculate how many or what type of genotypes will be there. Crosses, we have studied about all these type of crosses: reciprocal cross, back cross, test cross. So what happens in the reciprocal cross? If we are reversing the sex, suppose in one case the parent is having this particular combination: capital T, small t, small t. So suppose the male is capital T and the female is small t, small t. But if in another cross, okay, we make the female as capital T and the male as small t, that would be basically reciprocal cross because we are reversing the sexes of the parents. So this is reciprocal cross. What do we do in the back cross? Whatever generation is obtained. So suppose capital T, small t fuses to form this thing, capital T, small t. Now, if this capital T small t is fused either with capital T or with any of the parent, with any parent, then that would be called as a back cross. Okay, so this would be called as a back cross. Now, talking about the test cross, if we have the parent capital T, small t, F1 generation is capital T small t. And if this capital T small t is fused with the homozygous recessive parent, okay, only homozygous recessive parent, then this type of cross will be called as the test cross. Yes? Monohybrid cross, this is an example of monohybrid cross, where only one characteristic is considered or one trait is taken. Dihybrid if two traits are taken. Yes, monohybrid, dihybrid cross. If the two traits are taken, we have already studied this in detail. Anyway, we look at what is dihybrid cross. But when two characteristics are taken, then it will be a dihybrid cross. Okay. Moving further, talking about the Mendel's monohybrid cross in detail. So in monohybrid cross, as we talked about, what we have to remember, there will be one pair of characteristics. Okay, contrasting character. In this, what we have taken as one pair, we have taken the height. Yes, height of the plant is on. In this particular condition, in this particular example, the condition that we have taken is color of the flower. Color of the flower. Either the color of the flower is purple. Yes, that is a dominant trait, or the color of the flower will be white. So that is small p and small p. Okay. Phenotypic ratio will be 3 is to 1. Genotypic ratio will be 1 is to 2 is to 1. How is the phenotypic ratio 1 is to 2 3 is to 1? Because we will see that actually three of the plant are purple, three of the flowers are purple and one is white. And what is the genotypic ratio? In the genotypic ratio, we'll see that it's homozygous tall and homozygous dwarf. So homozygous tall is this one. Okay. Homozygous heterozygous tall will be capital T small t, and homozygous dwarf will be small t small t. If you're looking for this example, then it will be purple. Okay. Yes. This will also be heterozygous purple, and then this will be white. Yes. If you're taking this particular example, but Mendel did it with the height, so we are looking at both of them. Law of dominance came from monohybrid cross. What does law of dominance say? Law of dominance says that in a heterozygous condition, the allele whose characteristics are expressed over the allele is called as dominant. So basically, in this condition also we'll see when the fusion takes place, then looks like that all the flowers are purple. But are actually all the parents purple? No, one parent is white. That means that one characteristic is expressed over the other one. And this particular is called as the dominant one. Yes, dominant allele. So this was about law of dominance. But after a while, we saw when the selfing was done. Suppose if the Mendel had taken capital T, small t as the characteristics, the first generation which came was all plants were tall. But in the second generation, what did we observe? We observed that there was one homozygous tall. Then we observed that there were two heterozygous tall, and one we observed was homozygous tall. Yes, so that means that the recessive characteristic is coming now. That is, it is not lost. The two factors of the character present in an individual, see, do not get mixed up. This is what you have to focus on over here. See, they do not get mixed up, and both the traits are recovered in the F2 generation. See, in the F2 generation, both the dominant as well as the recessive traits are recovered. Yes, is that clear? Now, moving on to the next one, that is the Mendel's dihybrid cross. So, dihybrid cross means that we have taken two contrasting characters. Yes, we have taken two contrasting characters. And what are the two contrasting characters we have taken? We have taken here the yellow round, okay, yellow wrinkles, okay. So we have taken the yellow seed color, it will be rounded wrinkle, then green roundabout wrinkle in the ratio of, and the ratio that we obtained is 9 is to 3 is to 3 is to 1. Okay. So how do we do it? We have already studied about the Punnett square. So we have taken two characteristics over here. That is why it is called as the dihybrid cross. Yes, two characteristics were taken, and when these were fused, these were the gametes that we have observed. Yes, so how were the parents looking like? See, the parents look like this. Capital A, capital A, capital B, capital B. Yes, into small a, all recessive. This is the gamete that we obtained. Now, when we did the selfing of the gamete, in F2 generation, these are the genotypes that we will observe. So, these are the genotypes that we have kept over here. Yes, these are all the genotypes kept over here. One is supposed to be male and the other is supposed to be female. Okay. So, what was the result that we obtained? The result that we obtained was that round and yellow. See, you can see here round and yellow. How many are there? 1, 2, 3, 4, 5, 6, 7, 8, 9. So, the round and yellow that we have obtained are basically how many? 9. If we talk about wrinkled and yellow, okay, so if we talk about wrinkled and yellow, what do we see in wrinkled and yellow? There is 1, 2, and 3. 3 wrinkled and yellow. Then if we talk about round and green, how many round and green are there? 3. 1, 2, 3. And how many finally the recessive one? What is the recessive? It is supposed to be green and it is also supposed to be wrinkled. So that is the recessive one. That is 1. So this is the final ratio that we obtain in the Mendel's dihybrid cross. So after from the Mendel's dihybrid cross, we also study the law of independent assortment. So what does the law of independent assortment states? Look over here very closely. When two pair of traits are combined in a hybrid, segregation of one pair of character is independent of the other pair. See what is separated here? Here in F1, everything what we obtained was round and yellow. But we saw in the F2 generation wrinkled and yellow also, green and round also, green and wrinkled also. That means all the characters are independent. The character is independent of the other pair of characters. Okay. Now moving on to the exception to Mendelism. So this was all about the Mendel where we studied the monohybrid and the dihybrid cross. Now what all is considered as the exception to Mendelism. Moving on to incomplete dominance. Now, incomplete dominance is important. Here you can see what did Mendel say? That what is dominant will express itself in the F1 generation. But in this example given in your NCERT, antirrhinum majors, okay, you can see that this particular flower is a red colored flower. Then this is a white colored flower. So these were present in the parental generation. When they fuse, what should be the F1 generation? According to Mendel, the F1 generation should be red. But F1 generation all came out to be pink. That means both of them were incompletely present. Even the red was incomplete, the white got incomplete. That's why they were not, both of them were dominant. Okay, so red was also slightly dominant, white was also slightly dominant, hence incomplete dominance. So this is one exception to Mendelism. Okay, now moving on to the another exception to Mendelism is a co-dominance. So in incomplete dominance, both were dominant. Okay, both were trying to be, express actually, and they were not able to express themselves very nicely. In co-dominance, and both are expressing very well. They are not blending. Okay, they are not blending, unlike the incomplete dominance. So suppose if this is the cattle color and here is another roan color cattle. So we can see that the cattle which is obtained in the next generation is there would be spotted cattle. So white color, cream color cattle coat having roan color cattle coat also. So they are independent of each other, both of them expressing themselves, not also blending. Now, moving on to the next one, that is the multiple allelism, which is another exception to Mendelism. Mendel said that each gene has only two alleles. Yes, but the multiple allelism says that this is wrong. Sometimes you can see that for a gene, many alleles are there, as you can see in the case of the blood groups. In the case of blood groups, you can see a little more. The alleles can be, see, A for blood group A, two alleles are responsible, IA, okay, IA, for B, IB, for O, simple I. There's, nothing is present in that. So for blood group, there are three alleles. IA is also there, IB is also there, and small I also you can, denote it by simple I or I. Okay. So Mendel said that there are only two alleles, but we found well there could be multiple alleles. So these were the exceptions to Mendelism. Okay. Now moving on to and studying about the chromosomal theory of inheritance. Because when we found out about the exception to Mendelism, we got to know that okay, there could be, there is chromosome, we should work on the chromosome, and actual gene of inheritance is basically this particular chromosome. Now, chromosomal theory of inheritance states that during the meiosis, that is during the reductional division, we will see that the homologous chromosome pairs migrate as discrete structures that are independent of other chromosome pairs. Now, in 11th standard, when you have studied the meiosis, you have seen that after the prophase one, when the chromosomes have undergone the chiasmata is there, they have exchanged the information. After that, you will see that they will independently move. Okay, whenever you will see that there is the attachment of the spindle fibers, they will independently move of other chromosome pairs. This sorting of chromosomes from each homologous pair into pre-gametes appears to be random. And this process is completely random. Okay? It could be anything. That's why we have so many gametes, and so many gametes have different set of and different arrangement of chromosomes. So this is very random. Each pairing synthesizes gametes that contain only half of their chromosomal complement. Yes, in the humans also. We have 46 chromosomes. Yes, but in our sex cells or in our gametes, that is the ova and in the sperm, how many chromosomes are present? Only 23 and 23 chromosomes are present. So only half of the chromosomes are present in the gametes. Even though male and the female gametes, sperm and the egg, differ in size and morphology, they have the same number of chromosomes. See, ova is very big, sperm is very small, but even though due to the difference of size, you will see that the arrangement of chromosomes is completely in both the cases. So that there is equal genetic contribution. If it is not equal, this genetic contribution will not be equal. Now, the gametic chromosomes combine during the fertilization to produce offspring. Yes, this is what will happen, right? So, there is one set of chromosomes, C will come from the sperms, and one set of chromosomes, red color over here, will come from the ova or the ovum. Yes, and both of these will then join. Homologous chromosome will be formed. The meiosis one will take place where there will be synapses. Yes, they will exchange the information. There will be chiasmata formation, and then after that, the daughter nuclei is formed. So basically, the exchange of information takes place. Yes, is that clear? On this, the law of independent assortment is used. So, law of independent assortment states the same thing, that the alleles of the different genes are inherited independently within the organism that reproduces sexually. So it is independent assortment. All the genes, all the alleles, all the chromosomes will independently move without any pressure or without any influence from anyone. So it moves without any influence. Yeah? Okay? Now moving on to the very important topic, linkage. So linkage was first time seen by Betson and Punnett in Lathyris odoratus, which is the writing. What does linkage mean? See, anyway, linkage, if you're sitting with your partner, you share your lunch, you go to school together, there's a linkage. So there is a close association of genes or other DNA sequences on the same chromosome. So if two people, they are neighbors, definitely there will be close association. And the closer two genes are to each other, the greater is the probability that they will be inherited together. The closer you are to your neighbor, the greater is the probability that you will be in the same class, in the same section because you are good friends. Moving on to recombination. Recombination will be basically recombining. And when will this recombining take place? There will be breakage of chromosome. Chromosome breaks, there will be separation of genes as we saw in the independent assortment. Okay, so suppose your capital A is there, okay, and then small a is there, this will break, and the capital A, might go on another segment of small a, alleles will break. Genes will also break. Suppose in this chromosome, here is the gene for eye color, and here is the gene for hair color. And mostly they go hand in hand. But sometimes we will see that they will also get exchanged. Maybe in the next generation, this eye color gene goes to separate person, and this hair color gene goes to separate person. This is called as the recombination. Formation of the genetic combinations in offsprings that are not present in pairing. This is not present in pairing. This is the pairing. This has both the genes. But here in the offsprings, F1 generation, we see that both the characteristics are now separated, So here also you can see in the recombination scene, this is one chromosome, then there's chiasmata formation. Okay, they are exchanging their genes, their traits, and after the exchange, you can see that they have separated. They have separated. So this is what the independent assortment and the recombination is teaching us. Genetic maps. Now, genetic maps are constructed for us to actually see the arrangement of genes. We should know where all the genes are. So, they are extensively used as a starting point in the sequencing of whole genomes, as was done in the case of human genome sequencing process. So, what we need to know, this is the chromosome. Right over here, you can see we should know everything about this chromosome. On this location, there is this gene which is responsible for eye color. Okay. On this particular location, there is this gene which is responsible for the wings. On this particular location, there is another gene which is responsible for the legs. So that is why genetic mapping is very necessary. Okay. To know the location and to know the characteristics of the gene. Talking about the sex linkage. So linkage we've already studied. Sex linkage means we'll talk about only those genes which are linked to sex chromosome. What are the sex chromosomes? X and the Y. So, and how can we calculate the linkage group? So the number of linkage group is equals to the haploid number of those homologous chromosomes. So haploid number of those chromosomes will be, if you talk about X or the case of Y, what will be the haploid number of homologous chromosome in this case? 23. So the linkage group that we see will be 23. The genes which occur exclusively on the X chromosome are called X-linked genes, and which are present on the Y chromosome are called as Y-linked genes, or they are also called as the Holland, because they carry the male characteristics. So male characteristics are also called as the Hollandic genes. Now talking about examples of sex inheritance in humans. See, there is inheritance of X-linked. So X-linked means some genes will come only from X chromosome. So some diseases that we see, color blindness, hemophilia, only present of X chromosome will be transmitted from one X chromosome to another. Inheritance of X-linked dominant genes, there could be X-linked dominant genes like hypoplasia, which is also present of X-linked, this dominant, hence carried on to the next generation. Talking about the next topic, sex determination, we know very easy. In humans, we've studied many times. XXXY is the sex determination system that we see in the case of the male humans. In the females, it is XX. In the males, it is XY. Yes, so there is 44 plus XS, 44 plus XY. And the gamete which will be formed in the case of female, all gametes will be X. But in the case of male, all gametes will not be X, but in fact there will be 50% X and there will be 50% Y sperms. Yes? Moving on to the next method. In the case of birds, we will see that the females are basically ZW, okay, and the males are basically the homogametic one. Whereas in the case of human, the females was homogametic. Okay, but in the case here, we'll see that the female is a heterogametic one. So ovum is the one which will determine the sex of the offspring. Whereas in the case of humans, sperm determines the sex of the offspring. Yes. Now moving on to the sex determination in bees, there is the XX exosystem. So what do we see in the XX exosystem is that the female is basically, again, it is the homozygotic over here, and the male is going to be the heterozygotic. So O does not mean, O means that there is nothing over here, it's empty. Okay, so this is the heterogametic sex over here. In this system, there is only one sex chromosome referred to as X. Males have only one, females will have two. So here we can see in the case of the bee, XX exosystem. Yes, the zero on the O system signifies that there is the lack of a second X. Now, moving on to talking about mutation. Mutation meaning the changes or the alteration in the DNA sequences, okay? And these changes that are inheritable. So we can also say that the mutation are basically inheritable changes. Yes, they are inheritable changes. Resultant alteration of DNA sequences leads to genotypic and phenotypic variation. There is change in gene, and because of the change in gene, there is also change in the phenotype. Some mutation takes place, and maybe our eye color is different. We are heterochromatic. One eye color is black, one eye color can be blue. Two types, it could be point mutation, frame shift mutation. What is point mutation? Suppose if there is, a mutation which is taking place at only a certain point or a certain, uh, nucleotide is changing, that would be called as the point mutation. But because of the point mutation, there can be frame shift mutation. Suppose A G C T. Okay, this is DNA code. But if there is a mutation that took place, and how do we read the gene like this? Triple A, GCG, CTA. Okay, we'll read the code like this. They are very different. But suppose there is a mutation at or there is a addition of a nucleotide somewhere. So if the addition is taking place, then suppose T got added here. So it will be AAP, then AGC, then GCT. Okay, so here we'll see that basically the reading, the whole frame is changed. That's why this is called as the frame shift mutation. And the agents that will cause this mutation are called as mutagens. So there can be UV radiations or infrared radiations which could cause these mutations. There could be certain chemicals that can bring about the mutations. So they are called as the mutagens. Now moving on to a very important topic, that is the pedigree analysis. So pedigree is basically, it is used for us for the reference or to maintain a family history of a tree. Suppose you must have seen that if your father has diabetes, then it is very likely that the son will have diabetes. So diabetes is a disease that runs in the family. And if you want to track this disease, you have to create a whole tracing map, right? So this is all done by the pedigree analysis to analyze the family's history of the trait. What all can pedigree tell us? It can tell us that the trait is dominant or it is recessive. It can also tell us that it is which type of chromosome. Is it the autosomal chromosome or a sex chromosome? It could also tell us about the trait which it is linked to. It could also tell us about the genotype of the family members, and it could also tell us about the phenotypes, how we will look in the future generation. And how many types of these can be. It could be autosomal dominant. Autosomal means it is present on the 22 chromosomes, not on the 24 sex chromosomes. It could be present on autosome, it could be dominant, okay, or it could be present on autosome and it could be recessive one. It could be present on X-linked, X is that is present on the X chromosome, and this also could be dominant, and it could be present on X chromosome and it could be recessive. Yes, so this is how the pedigree analysis tells us. There are symbols that we study for pedigree analysis. You already studied. You already know that the round shape tells us that it is a female. The rectangle, the square tells us that it is a male. And if it is mentioned in this particular rhombus structure, that means that the sex is unknown. Then if it is effective, then it is going to be shaded. If they are siblings, then they are attached by this link which is coming from the parent. Suppose the parent have mated and they have produced 2 or 3 babies, then this kind of link will be there. But if the link is between two individuals, that means that they have mated, they are mostly husband and wife, or they have mated. Consensuous mating means if the mating takes place between the sisters and the brother, between the relatives, then this is supposed to be consanguineous mating. That is why it is expressed with 2 lines in between. Okay. So the pedigree analysis that we'll study, we'll see first autosomal recessive. So autosomal recessive, both can be carrier or one can be carrier. But if only one is carrier, then the child won't be affected. But if both the parents are the carrier of the disease, then one child is going to be affected for sure. Two child can be carrier and one can be unaffected. So this is when it is present on the autosome and the disease is recessive. But if we talk about the dominant case, so dominant case we will see that there are basically no carriers over here. Okay, either the disease will be there, so either the disease will be present, yes or no. There is no carrier here. But in this case, you saw that the carriers were present. Yes, so either the child is going to be unaffected or he or she is going to be affected. So this is about the autosomal and it can happen to any sex. Okay. But in the case of the X-linked dominant, okay, so in the X-linked, we will see that that particular disease will mostly, if it is in a father, then it will only be affecting the only affecting the daughters. Okay, if father has it. Okay, but if it says that the mother has it, then mother can give it to both the sons and the daughter, both the son and the daughter. But the daughter is mostly going to be, unaffected. It could be a carrier daughter also. So mostly the daughter is going to be a carrier daughter. Okay, so this is going to be the X-linked disease, X-linked dominant. Now, talking about the coming to the end of the chapter, we have already studied about the chromosomal aberrations. We are anyway, just going through and summarizing it. Chromosomal aberration. Aberration means, anything which is wrong, anything which is, unusual. So, what is the unusual thing that we see in the chromosome? There could be addition or deletion of chromosome. There could be 46, so there could be +1, that is 47. There could be -1, so there could be 45 chromosomes also. So addition or deletion of chromosome can take, duplication of the whole chromosome pair can take place, failure of separation of, because of the segregation of aneuploidy. So basically, ploidy would be the chromosomal aberrations. It could be of two types, euploidy or aneuploidy. In the case of euploidy, we will see that basically the whole chromosome pair is doubling. The whole chromosome pair is doubling. Whole chromosome pairs, double or they are not able to segregate. Aneuploidy, there could be change or alteration in only one chromosome. In one chromosome. Okay. So this can be hypoploidy or hyperploidy. Hypoploidy means the chromosomes are getting less. That -1 chromosome or -2 chromosome. Hyperploidy means addition of one chromosome, addition of two chromosomes. Whereas in euploidy, there is addition of whole set. The whole set of chromosome is added. Whole set is added. Okay. So +1, +2, hyperploidy. Hyperploidy case will see monosomy. Suppose if the base there were 2 N chromosomes. If one is gone, monosomy. Two are gone, nullisomy. Trisomy is when one chromosome is increased, and tetrasomy is when two chromosomes are increased. Okay. So we are going to study about the cases of this one. Down syndrome, you must have seen this in some people around you. Abnormal cell division is taking place and there is an extra or full or partial copy of the chromosome number 21. So basically there is extra chromosome number which is present at the 21st position. 21st position, there are supposed to be only two chromosomes, but there is another third one which is come. That is why we say that there is trisomy at 21st position. Trisomy at 21st position. What will be the characteristics? Round face, you can see over here, broad forehead, short neck, the tongue is going to be furrowed, Mongolian type eyelids will discuss. Okay, this is called as the Down syndrome. It is also referred to as Mongolism. Mongolism. Now moving on to the next one, that is the Turner syndrome. The Turner syndrome means there is monosomy at 23. So 23rd chromosome, where it is supposed to be that two chromosomes should be present, in turn one chromosome is gone. So there is only one chromosome that is present at the 23rd position. And this mostly takes place in the case of females. So in females, if one chromosome is gone, that means there would be less feminine characteristics because only one X chromosome is present. So the breast will be undeveloped, uterus will be small, ovaries will be sterile. So basically, less feminine characteristics is what we'll see in the Turner syndrome. Now moving on to the next one, that is the Klinefelter syndrome. In Klinefelter syndrome, what we see is that on the 23rd chromosome, there are basically 23rd position, two chromosomes there. Okay, there is going to be additional X chromosome present. Okay, so additional X chromosome is present. That is why the total number of chromosomes now are 47. Okay, here in these organisms, in these cases, we see that they have undeveloped testes. Testes is not very well developed. Mental retardation is there. Knock knees are there. Feminine pitch voices are there. These are the people that are present in the society and they're called as the transgender. Okay, because they can't actually scientifically qualify into the 46 chromosome as male and females. Baldness is there, poor beard growth is there, the shoulders are narrow, tendency to grow few chest hairs, breast development is there, but it's not very well developed. So there is basically less breast development. Females that pubic hair are present, there could be wide hips, small testicular size, and there will be no arms and legs. So this is all about the summary of this chapter. We have very quickly gone through the whole chapter. So please study this chapter as it is very important as per the outlook of your NEET as well as the board exams.